Hey guys, welcome back. Here we are with sections 3 and 4 of chapter 11. Remember, chapter 11 is gene expression and epigenetics. So to review, epigenetics is when there can be a change to how or when or how often genes are expressed, but it's not an actual change to the DNA. It's a change to these chemical groups that are added to the top of the DNA or to the histone proteins that the DNA is wrapped around. Now, since it doesn't actually affect the DNA coding, then that means that it is not permanent. It can be reversed. But until it's reversed, it can also be passed on to the next generation. Now, we talked a lot about what it was and how it worked in terms of the chemical groups, how acetyl groups will, when they attach to histones, it causes the histone proteins to contort, loosening up the DNA and allowing transcription factors access to the genes so they can transcribe them. And if you have methyl groups added, it has the opposite effect. It actually causes the DNA to wind up even tighter, making it near impossible for transcription factors to access those genes and thus silencing those genes, preventing them from being expressed at all. Then there's also the phosphate group, which kind of does a little bit of both depending on the actual circumstances and other variables. Now, sections 3 and 4 are more so related to things we've actually already talked about, because since this is about gene expression, this is going to be very similar to things we've talked about in previous chapters. A lot of it's going to be almost identical, so that will make this part nice and easy, which is why I kind of combine them all into one. All right, so without further ado, let's move on to chapter 11, sections 3 and 4 notes. Yeah, no, there we go. Aha. There we go. So maximizing genetic information. Again, we've already kind of talked about this, but if you remember, as we said, most of your DNA doesn't actually code for proteins. So the small amount that does code for proteins, we have to be able to get as many possible proteins out of it. And how we do that is by differential intron splicing. And what that means is, like when we talked about RNA processing, you cut the introns out and then you're left with the exons, you squish the exons back together. And that's how you get the processed DNA, or sorry, mRNA to now be transcribed into a protein. But if you cut out different introns, different sections that maybe were considered exons previously, but are now considered introns, and things that were introns previously are now considered exons, you have a new group, a new sequence of bases, and that means you're going to have new codons. When you have new codons, that means you're going to have new mRNA and new amino acid sequences. And if you have a new amino acid sequence, then that means that when it comes time for protein folding, it's going to fold up in different ways and create a different protein with different functionality. Exon shuffling is exactly what it sounds like. You know, when you take the cards, you shuffle them together. Well, instead of taking out, let's say, this, and this intron, and then just pushing the exons together, what if we move the exons around first? When you do that, you might have the same bases present, but they're not in the same order. And since they're not in the same order, that means we're going to have different order of amino acids. And if we have different order of amino acids, it means we're going to have different folding to our protein, which means it's going to have a different function. So these are ways we can get different types of proteins being made from the same gene. So we can maximize our genetic information by rearranging the mRNA in order to create different different proteins from it. And that allows one gene to produce multiple proteins. This is how your body can make well over 22,000 different proteins with such a small amount of DNA. And I'm not saying your DNA is a small amount. I'm saying that the amount that codes for proteins is a small amount. We've talked about this, 1.5 to 2 at most %. So going back to the idea of the differential intron splicing, so intron sequences of one gene can contain the coding sequences of another. Just because I take out introns 1, 2, so here are my sequences: exon, intron, exon, intron, exon. Just because I take these two out, no, I still have fingers up. Just because I take these two out and then put the middle finger and the thumb and the pinky together. I know it looks really bad, I'm sorry. But just because we do that doesn't mean that to make another protein, I can keep these two introns in, but I'm going to take out those two, what were previous exons, but now introns, and now I can make a whole new protein out of it. Or I could, let's go back to this one for a moment. So that's what it means by differential intron splicing. Or I could rearrange my fingers, and we have the pinky in the middle, new order, new protein. So that would be the exon shuffling. Now at the end of this, I do have a link to the to the Prezi, and you'll also notice that there are additional resources. One of them is a video made by me that talks about RNA processing and goes into differential intron splicing and exon shuffling and all that. I would definitely recommend checking it out. I use my whiteboard and I diagram it to hopefully make it a little easier to understand. All right, let's check out the picture. So this is kind of saying, all right, well, in one, let's say all of these are together. So we have all of these were originally together, but we take out A, B, and C, and we're left with 1, 2, and 3. That becomes a gene. Or maybe we take out 1, 2, 3, and we're left with A, B, C. Well, that's another gene, or sorry, that's another protein. Or we can leave in 1 and 2, and B and C, take out A and 3. Well, now that we have different bases, it's going to be a different protein. Or what if we just have A and 3? So we would have a different protein from just that. So there's ways to start off with the same original mRNA sequence and have different ways to process it in order to have different proteins. All right, section 4. Most of the human genome actually does not code for proteins. Dun, dun, dun. I know this sounds like a big shock, but we've talked about this before. There we go. Put this back up. So about 98% to 98.5%, somewhere around there, actually doesn't code for protein. Only 1.5 to 2% of your actual DNA codes for protein. So that leads to the question of, well, what are the other functions? Now, again, I know we've talked about this in the previous chapter, but we're going to talk and review about it a bit more. I'm also going to add some new stuff to it. So the other portions. Now, as we've already mentioned and previously, the part that encodes for genes, that's the part that gets transcribed into the mRNA. But what about the rest? I mean, we've talked about rRNA, the ribosomal RNAs, or the tRNAs, or the snRNAs, or RNAis, snORNAs. I mean, there are so many different kinds of RNAs, and we have to be able to make them from the DNA as well. So a full third of our genome actually codes for RNAs that are different than the mRNAs. So the mRNAs are encoded in that 1.5 to 2% that code for proteins. But all of the other RNAs, they have to be encoded for as well. And they're located in the non-protein encoding section. So a full third of our genome actually codes for all the other RNAs. We've mentioned pseudogenes before. These are genes, or well, they're portions of the DNA that may or may not be transcribed. If they're transcribed, they're never translated into a protein. Not 100% sure on what the function of pseudogenes are, but we do know that there are occasional disorders that occur if a pseudogene crosses over with a functional gene during meiosis. We'll get to that in chapter 13, I believe it is. That sounds about right. Chapter 13. That's on chromosomes. All right. And point 3, a large part of our human genome is composed of these repeated sequences such as signs and lines. Now, we've talked about signs and lines in the previous chapter. And we said that the signs are these shorter but sequences and the lines are the longer sequences, but they're just repeating sequences of bases. And as we've mentioned before, we don't really fully know what these are. These are going to be part of the telomeres, which we're going to talk about later. But not just that, these are also, it's believed to allow for less chance of mutations occurring in a protein encoding section and greater chance or greater probability of a mutation occurring here. It's essentially a game of probability, a game of numbers. If you have this much DNA and only this much codes for your genes and your proteins, then a mutation is more likely to occur in this other region. So that's one thought, that it could just be these buffer zones that allow for a likely place for mutations to occur rather than to occur within something that encodes for proteins. All right, let's see. Pew, 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 pew. Pew, 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 pew. Pew, pew. And last but not least, viral DNA. Yep, you got some virus in you. You got a virus in you. All right, sorry. So we actually have found different viral sections of our own genome. So that means that we ourselves have actually incorporated viral DNA and viral RNA sequences into our own genome. Roughly, they think it's about 8% of the genome, about 100,000 sequences within our DNA were actually once part of RNA viruses. Now, if it's an RNA virus, we call it a retrovirus. And that just means that instead of having DNA as their primary genetic code, they have RNA instead, just RNA. Now, it's just kind of interesting to think of this because a lot of times when we think of viruses, especially now, because I know this probably won't hold up in the future, but right now we are going through the COVID-19 pandemic. And so right now we're thinking about viruses and how they're always bad and horrible and, disease and all that, which, for the most part, starts off true. However, there are ancient viruses that left their imprints in our ancestors' DNA, and it's remained there ever since. Now, there's a lot of studies on these to see what kind of effect they could have. And I actually have this great article that I'll probably share with you guys at some point in class, but it talks about how these certain sequences that are viral in origin, but has just been left off in our genome and been continued to pass on through the many, many generations, actually helps with mom's body during pregnancy. It actually helps her to maintain a healthy pregnancy. So it's just kind of interesting seeing how, not all bad. Sometimes it can be good. Anyways, we call these HERVs, HERVs, and that stands for human endogenous retroviruses. So these are those RNA type viruses that long time ago, they infected our ancient ancestors and left behind some of their genetic material that was just kind of incorporated into the DNA of our ancestors. And since it was incorporated into their DNA, it got passed through the generations, through the many generations, and we still have them today. So just kind of something a little cool if you really think about it. I know I'm a nerd, I'm a geek, I'm a dork, but that's why you guys love me. All right. And other portions we already talked about. Those are like the signs and lines. And so that brings us now to additional resources. As part of this lesson, I would definitely suggest you checking out some of these videos to help cement your understanding of this. Now, these additional resources are not just about sections 3 and 4. They're about the whole chapter. So with Science in Pajamas, I did an epigenetics video to kind of talk more about what nucleosomes and histones and DNA are and how these different chemical groups affect them, and thus how that will affect gene expression. So it might be a good one to check out. Also, RNA processing. We don't just talk about the basics of processing, such as the modified nucleotide cap, the poly A tail, and the intron splicing. It also does go into differential intron splicing, exon shuffling, and things like that. So might be a good one to check out. Very visual. Could definitely help. And then there's also these other videos. So we have one by Bozeman Science, and that talks about epigenetics in terms of nature versus nurture. So very informative on how it affects gene expression, but doesn't affect the DNA. We have a TED educational talk about what is epigenetics. Again, very informative on the subject of epigenetics. And then we have McGraw-Hill Animations, which looks at exon shuffling. So again, this is kind of looking at the idea of getting the most out of our genetic material by using the same DNA gene sequence to make multiple proteins. So again, just some more information to help with that. If you guys have any questions, definitely hit me up in our Google Classroom, send me an email. But I hope this was a good lesson. I hope that it helped. Definitely check out some of those videos. They are very informative and they will help with your understanding. They have a lot of good visuals as well. But until next time, everyone, take care of yourselves. Stay safe, stay healthy, and I will talk to you later. All right. Bye, guys.